go over the concept of allosteric regulation we see in enzymes. Remember that the allosteric site is the region away from the active site on an enzyme that's going to affect the active site in some way. Whenever a certain molecule binds with this allosteric site, it's going to change this active site in some way. But again, this allosteric site is away from the active site on an enzyme. You can see our enzyme here is going to bind with the substrate because the substrate matches the shape of the active site of the enzyme. Now, there are two ways these allosteric effectors affect enzymes. The first is by activating the enzyme. Whenever the allosteric activator molecule binds with the allosteric site of the enzyme, it's going to change the active site in some way. The interaction between the allosteric activator and the enzyme is going to activate the enzyme to function properly. You can see our enzyme here has an allosteric site down at the bottom that the activator molecule is going to bind with. It's going to change the shape of the active site of the enzyme. You can see the shape here is different from the original enzyme. So again, this is going to activate the enzyme. The shape of the substrate probably looks like this half circle compared with this half circle, because this activator is activating or turning on this enzyme. The other type of allosteric regulation is inhibition. This is when a molecule is going to bind to the allosteric site and actually inhibit the enzyme from functioning properly. We can see our inhibitors here on the sides of the enzyme, changing the active site of the enzyme so it won't bind with the substrate. This again will stop the enzyme or deactivate it from reacting with the substrate.